Lifesciences Incorporated is a pharmaceutical company engaged in the distribution, importation, registration, and marketing of various therapeutic products across the Philippines. We at Biocare commit ourselves to service excellence in the promotion and delivery of essential as well as technologically advanced medical products that are within reach of the average Filipino. We pride ourselves in our company core values: punctuality, integrity, commitment, accountability. And this reflects on our service. Our vision is to play a significant role among the pharmaceutical companies in the Philippines, delivering high-quality yet affordable products and services that improve the quality of the Filipino lives it touches. We are dedicated to our mission to be among the top market leaders in our chosen segments by providing qualitative and affordable medicines to the community and year-to-year growth of employees and stakeholders, as well as to develop a young, talented, and experienced workforce. In 2011, Biocare Lifesciences Incorporated was founded by Managing Director Dilip Tiwari and President Kimberly Chua Tiwari. By 2012, we started the distribution of our products to numerous multinational and national companies. In 2013, Biocare registered over 23 products for different segments, and by 2015, we expanded our staff, deploying our new sales force to 17 areas in Central Luzon, South Luzon, and the Visayas region. In 2016, the expansion of the sales force and marketing team continues to the Greater Manila area, Visayas, and North Luzon, and we've moved to the new HQ with a complete rebrand. By 2017, we successfully launched Sepsul, a combination of third-generation cephalosporins and beta-lactamase inhibitor antibiotic. Now, Biocare expands to Cambodia and launched our first inhaler line with salmeterol fluticasone, Salflosol MDI, and Highcare, brand of salbutamol. Biocare has products from reputed pharmaceutical manufacturers around the world, whose facilities are approved by various regulatory boards. And Biocare hasn't even reached its full potential. In 7 years, we've only laid a foundation. Soon, we'll strengthen our reach to the Mindanao region and eventually spread to the whole Asia-Pacific region with a culture of leadership and accountability. Looking forward, we want Biocare to be a corporation that you can always trust. Providing quality, defining life.
Good evening, doctors. I am Paulina Espiritu from MedConcept, and I will be your host for this live webinar. To formally open this forum, let us welcome the Managing Director of Biocare Lifesciences, Mr. Dilip Tiwari.
Hi Doris, very good afternoon. Good evening to our doctors. It cannot be denied that the number of active cases keep rising day by day. Our healthcare system continually challenged and our economy greatly affected. As a healthcare partner, we are obliged to make available treatment modalities that will support you in management of COVID-19 transmission. Tonight's webinar will be an opportunity to present the use of immunomodulator for the protection against COVID-19. We thank you for accepting our invitation to attend this forum. With that, I would like to invite on a screen the Associate Vice President, Asia of Cadila Pharmaceuticals Limited, Mr. Paresh Tripathi, for his message. Thank you.
A very good evening and greetings from Cadila Pharmaceuticals Limited to all the esteemed speakers, panelists, and all the delegates from Philippines, India, and around the world who are participating in this event. Thank you so much for being here. To start with, okay, we are going to discuss about SEPSIVAC this evening, the unique immunomodulator from Cadila Pharmaceuticals. Just taking you through the Cadila Pharmaceuticals overview. Founded in 1951, 70 years of journey, more than 9,000 employees, operating in more than 100 countries. With our manufacturing facilities being approved by whoso of the globe, like US FDA, UK MHRA, TJ, Australia, and so on. And while we do our own research, we have strategic alliances to add value for our R&D purposes. Some of them are mentioned here. I will not take much time so that we can move on to the scientific session faster. Also, strategic partnerships for manufacturing, distribution, and marketing of all our pharmaceutical products around the globe. As I said, we focus on research and development. That is one of our core values, and we are present across the value chain of pharmaceuticals. That is R&D, research, manufacturing, distribution, and marketing of the products. We also manufacture most of our own APIs. The journey in brief from Cadila Pharma from 1951 to 2020, various innovations for the first time in the world products, and now in 2020 we come out with SEPSIVAC, the immunomodulator. That is what we are going to discuss, SEPSIVAC, today. Just a brief mention about our founder chairman, Sri Indervadan Modi, and the care continues under our current chairman and managing director, Dr. Rajiv Modi. Thank you so much. And with this, I would like to invite Dr. Vipin Sethi. Just a brief introduction for him. He has been a critical care specialist working with Apollo hospitals for n number of years, and now working with Cadila Pharmaceuticals as our global medical advisor. Thank you so much. Vipin, over to you.
Thank you very much, Paresh, and a very pleasant evening to everyone. Kamusto, namaskar, hello. I'm your host and host for today's session. I'm going to moderate this session, ladies and gentlemen, and today we have a wonderful webinar on the role of SEPSIVAC to overcome this very grim situation, I can say, the world is facing today because of this virus called SARS-CoV-2 and creating a COVID situation. The world is today struggling to have a proper medication to treat this condition. However, the various medications have been tried and used by medical fraternity, but still the cases are rising. And Philippines is one example. Southeast Asia is going through a very critical condition as on today. And many countries are still suffering. Even India is still, we get about 40,000 cases every day. To overcome this challenge, Cadila Pharmaceutical has come up with a medication called SEPSIVAC. And this is the webinar today so that we can treat the mankind out of this COVID situation. SEPSIVAC, which is produced by a bacteria called Mycobacterium W, it's an immunomodulator drug to boost innate immunity. Developed by Cadila Pharmaceutical Limited in coordination with Council of Scientific and Industrial Research, Government of India. It was a project. And to give a preview on this, I would like more to talk on SEPSIVAC about our Executive Director for Research and Development at Cadila Pharmaceutical, Dr. Bakhilesh Khamar. So ladies and gentlemen, let us welcome Dr. Bakhilesh Khamar, please. Dr. Khamar, floor is to you. Over to you, Dr. Khamar, please.
Good afternoon, everybody. Good evening, everybody in Philippines. I'm happy to be here to share how we develop this SEPSIVAC. So I will take you through the journey of our development. So as you know, innate immunity is the first line of defense, and it is one of the fastest, and it recognizes all the organisms. It is broad spectrum, very specific. In spite of the broad spectrum, it's very specific and is consistent. You may know that all of us are getting infections, but we are not getting infected. So exposure and getting infected is different, and that is basically because of the adequate immune response generated by body. When that response is inadequate, we lose in the tug of war and we get a disease. When innate immune response is drastically low, there's a dysregulation of the response and there's organ dysfunction. So having said that, I'll take you through the journey of what we did in the preclinical studies. So we identified that it induces innate immune response by TLR2 and NOD2, while it induces potent innate immune response. It also antagonizes only TLR, which is TLR4, 5, 7, and 9, and they are mainly overexpressed in gram-negative sepsis and COVID-like conditions. That happens on the surface. Within the cell, signaling pathway, it downregulates P38, which is responsible for all the cytokines we see. Within the nucleus, at a transcription level, it upregulates all the genes which are upregulated in the sepsis and COVID and downregulates them. And then once the transcriptome is over, cytokines liberated by the cells, it downregulates all the cytokines, whether they are pro-inflammatory or anti-inflammatory, on its own, as well as LPS induced. And if this was not adequate, we took a next step, because all this happens in vitro. So whether it holds true in real life situation. So we induce pyrexia in the animals, which is known as the pyrogen test, and at the peak of temperature, we administered SEPSIVAC, and within half an hour, the temperature started coming down, while the animals which did not receive it continued to have a high temperature. Then we took a next step. Say fever is a small part of the whole game. Can we really make an impact? So we generated a model where E. coli induced gram-negative sepsis is created. Animals will die within 24 to 48 hours. And these are animals which are susceptible, organisms are susceptible to antibiotics. When you give antibiotic and steroid, which is standard of care, they survive a little bit. But when you add SEPSIVAC, there's a significant jump in survival. So essentially, SEPSIVAC gives you a multi-pronged approach. It works at multiple levels within the cycle of the sepsis. So now I'll take you through the clinical studies which we performed for approval of gram-negative sepsis. So first we did a dose finding studies where we had 3 dose level, 0.1 for 3 days, 0.2 for 3 days, and 0.3 ml for 3 days. All the injections are intradermal. And if you see on the right hand side, 0.3 ml given for 3 days had a maximum impact on a SOFA score, and so this dose was selected for taking it forward. And we did a next study to confirm these findings. So we had a 50 patients in ICU with gram-negative sepsis divided into 2 arms, and one arm received 0.3 ml of SEPSIVAC for 3 days. The other arm received 0.3 ml of placebo for 3 days. And as you can see on this slide, there is a significant reduction in hospital days from 16 to 10, mechanical ventilator from 9 to 6, ICU stay from 12 to 7, and all were highly significant if you look at the p-values. So having reconfirmed this finding that it leads to faster recovery, we went on to do pivotal study. And in the pivotal study, which is now published in CHEST, we tried to evaluate efficacy on a 28-day mortality. And the dosing regimen was same. And at the end of the study, what we found is that there was a significant reduction in mortality in those who received SEPSIVAC, which was 7.9% versus 18.8% in a control arm. This leads to absolute difference in mortality of 10.89%. And if you look into relative, it's a treatment benefit of 55.5%. And all this is highly significant. If you look at a p-value is 0.02, and then if you look at a number needed to treat, if you treat 9 patients with severe sepsis admitted in ICU, you will save 1 death. Not only that, it also provided more time for the drugs to work. So the death was delayed when you receive SEPSIVAC compared to when you don't receive SEPSIVAC. And if you look at a SOFA score over time, there was a significant faster improvement in SOFA score over time on day 7, day 14, day 21, as shown in the graph on my left hand. And we were also did a subgroup analysis to identify whether this effect is more in the less severe cases compared to high-risk patients. So if you can see on the graph on the right side, SOFA score, who has a higher SOFA score, more than 7, they had a maximum benefit in mortality rate compared to those who have a lower SOFA score. Because in the patient with lower SOFA score, as you know, less patient will die even in a control arm. So it holds true across the severity of disease. So summarize this finding, addition of MW improved 28-day mortality. There was absolute difference in mortality of 10.8%. Treatment benefit is 55% compared to the control arm. And if you treat 9 patients, you will be able to avoid 1 death. And all this is seen without any additional side effect. So the beauty of this product is it does not add any side effect whatsoever. It also delays time to death, which gives more time to maneuver and better manage the patients. And efficacy is better in severe cases. So it holds true across the spectrum of a disease. There's a faster recovery in SOFA score. There's a reduction in days on a vasopressor, and liver function, which was altered, also got better faster. Then, as I mentioned in the beginning, innate immunity prevents us from getting infections. So can we use this for a prophylaxis against respiratory tract infection? So what do we know about respiratory tract infection? So this viral infections of the respiratory tract requires TLR2 and not 2. And if you can control this, you can probably prevent getting viral infection, the respiratory tract infection. So as you know, I mean, I'm just reminding, SEPSIVAC is a potent TLR2 agonist and not 2 agonist. And then we take it through a trial where we took a cohort of 3,800 people and they received 0.1 ml of SEPSIVAC intradermally, and compared to those who did not receive, the protective efficacy was found to be 70.3%. But more important than this is that less number of people get severe disease. None of them had a severe disease requiring oxygen. Majority who get the disease were milder form. So if you take 100%, it's 78% will have a mild form of a disease when they have received 0.1 ml of SEPSIVAC. But this is in an ordinary citizens. What happens to the healthcare worker who are frontline and who are more exposed to this disease because they treat this patient? So in this study, 0.2 ml instead of 0.1 ml was given, which was followed by 0.1 ml 2 weeks later, and all this healthcare worker, frontline healthcare worker, were evaluated twice a day for 100 days, and incidence of COVID-19 in this group of patients was 32.3% versus 3.1% in those who received SEPSIVAC, which leads to protective efficacy of 93.3%. With this, I thank you everybody for patient listening.
Well, thank you very much, Dr. Kamar. I know that you have a dearth of knowledge about SEPSIVAC, and there is much more data which is available on the YouTube and on open web domain. But we'll come back to that, Dr. Kamar, once we have a panel discussion. But thank you very much for a wonderful, small, brief, wonderfully presented presentation on this. Now, with this, I move on to our next speaker, and let me introduce our first speaker for today evening, Dr. Abdul Samad Ansari, who is a practicing intensivist in Mumbai and is currently heading critical care for a large hospital in Mumbai called Nanavati Hospital. He was one of the early movers to join critical care as a specialty and has played a very important role in establishing critical care as a core specialty in India. He is a teacher, a regular speaker at various clinical forums, and he's a wonderful presenter. And I'm sure you would appreciate this once you start presentation there. So I request all the medical fraternity and all the doctors and my colleague to please join me welcoming live from Mumbai, Dr. Samad Ansari. Dr. Ansari, over to you, please.
Thank you, Dr. Vipin. Thank you for the kind words, and it's a privilege to be on this very beautiful forum, and especially love from Philippines. And it's a pleasure to be online with all of you. And with this opening remark and the kind of discussion that we had so far, I would like to take my presentation ahead. So as we have discussed that we are talking today about the sepsis and COVID-19, we'll talk about the correlation for clinical practice, and also we'll try to talk about the immune dysregulation in COVID-19 patients. So as Dr. Kamar very clearly and very nicely elucidated, I would like to go back for the benefit of the audience, that when we talk about immune dysregulation in COVID-19, very important to understand that we are talking about three distinct stages. The first one being the early infection, which is in the first 1 week where the viral response is very high, the virus is multiplying, and this is the time where you will see mild constitutional symptoms, dry cough, sometimes diarrhea, even headaches, body ache, myalgias. And if you look at the clinical signs, you will notice that there is a predominant lymphopenia coming in, rising D-dimer, LDH levels, very classically talking about evolving thromboinflammation. When we move to the second part, which is the pulmonary phase, which is again divided into two parts, where the viral response and the host inflammatory response kind of overlap, and the disease starts taking a kind of upward turn. This is where majority of the symptomatic people may start developing oxygen desaturation at rest or on exertion, shortness of breath, and a falling PF ratio. And if you do an imaging, you will find that there are abnormal chest imaging or CD findings. You will find there could be transaminitis, the liver enzymes going up, and there could be low or normal procal level at this point of time. So this is the most important phase once you enter the pulmonary phase, which actually starts from 8th to 14th day, and that dictates how the host inflammation and the viral interaction will lead to whether resolution or a stage 3, which is the feared one called hyperinflammatory phase, in which the patient will ultimately go into disorganized kind of immune response, ARDS, shock, cardiac failure, and this would be the fulminant point where the CRP, LDH, IL-6, D-dimer, ferritins go out of hand, the troponin NT-proBNP elevation happens, myocarditis sets in, and that's where probably the reversibility of the disease starts going down. Even if they recover from here, they go into a dysregulated state where there are secondary infections or fibrotic changes. Looking at the therapeutic options, as intuitively, antiviral role is maximum in the first phase, and this is the time where the antiviral like remdesivir or favipiravir and coupled with other antivirals can probably have some role. And finally, in this phase where you are hospitalized and are having pulmonary phase or hyperinflammatory phase, you need really immunomodulator. Now, going back, the discussion today is that can we have some kind of an immune enhancer at this point of time, which will also be immune modulator and immunotherapeutic. So both prophylactic and therapeutic action where the dysregulated host immune response can be manipulated. So in this quest, we will be talking about our therapeutic target today. Mortality in COVID-19, as you know, in mild and moderate cases is very minimal. Majority of them do recover, but in severe and critical ones, the hospitalization switching over to ICU is responsible for the death of the 15% of an odd number, 15% of the hospitalized serious patients. So out of 100 people, maybe 15 to 20% get hospitalized. Of that, nearly 15% means a hospitalized mortality of around 3 to 5% is what we are talking about, and a community mortality is still 0.05 or 0.1. So when we talk about COVID-19, it is just like talking about cytokine storm. Clinical features of patients were very clearly shown that there are pro-inflammatory cytokines and anti-inflammatory cytokines. The pro-inflammatory cytokines rise very early, and simultaneously there is a release of anti-inflammatory cytokines trying to get the balance and the harmony. But most of the time, when the inflammation goes unabated, later on a delayed anti-inflammatory stage can cause secondary superadded infection or reactivation of infections. So it has been very clearly shown very high cytokine level is correlated with mortality. So whether it is very high pro-inflammatory or anti-inflammatory cytokine level secretion, it is actually congruous with an increased mortality. So when we talk about controlling of the cytokine storm, has been very clearly shown to be beneficial to suppress these cytokine storm syndromes and immunosuppression, and that could be one of the interventions in early stage of COVID-19 pneumonia. So there was a discussion about growth factor reviews and something which can manipulate cytokine. And this is the area of interest. So when we talk about the editorial from John Vincent, which spoke about that when we talk about one size fits all approach, we actually need to individualize management. It was very clearly accepted that viral replication is actually coupled by inadequate host response, and therefore sepsis is an inadequate host response to infection. And this mortality from severe COVID-19 will be similar to any other form of sepsis. And when we look at the innate immune response to COVID-19, it is important to know that when SARS-CoV-2 virus with its spike protein, nucleocapsid, and envelope protein, and the genomic RNA invades, it binds the toll-like receptor and gets the activation, and that's called damage-associated molecular pattern recognition, or what is called toll-like receptor activation, leading to multiple cycles of activation of cytokines and chemokines, and sometimes overwhelming control, intracellular organisms, leading to cytokine storm, or in a delayed phase alveolar macrophage activation, causing a lot of hyaline membrane and kind of fluid accumulation and ARDS generation. If you compare, this is exactly what a gram-negative bacteria does by causing cytokine overexpression from the toll-like receptor 1 and 2 hybrid, and this is exactly the mirror image of a viral sepsis. So it was very clearly shown that in sepsis and other conditions like ARDS, out of hospital cardiac arrest, and trauma, the interleukin-6 levels, TNF levels, and IL-8 levels are always in an increased zone, 10 to 100-fold, 10 to 100-fold. And this is the mechanism of unexplained organ dysfunction. So when we talk about innate and adaptive immune responses against coronavirus, we must understand that our innate immunity is basically based upon non-trained immunity, which depends mostly about your macrophages, your monocytes, your dendritic cells, while the adaptive immunity depends upon the B cell, the humoral activation, and the memory cells, which can create antibodies. So innate immune system is the first line of defense, and this is the way in which we can strengthen an innate immune response to counter COVID from wreaking havoc over the first 7 to 10 days. And this is the therapeutic option that we are talking about, which can target immune deviation. So these three concepts of targeting the virus and its cellular life cycle by antiviral, or developing a vaccine that can prevent or mitigate disease symptom once the disease is already started to occur, and finally immune response motivation. And all of these things actually can be handled by SEPSIVAC to some extent. And this is how it works. If you look at this one graph, gives you an idea that whenever there is an activation of a macrophage because of SARS-CoV-2 infection or a gram-negative sepsis, there are 4 or 5 important things that can happen, which is SEPSIVAC can cause TLR4 antagonism, TLR2 agonism, or not 2 agonism, which is actually responsible for increase in the Th1 cell response. So if you can just go back to your immunity classes that we used to have in microbiology, that this is responsible for targeting the intracellular organism, while signaling changes and P38 downregulation, which is a mitogen activator protein kinase, these can all be beneficial in improving the interferon linked adaptive immunity control. And this is how it can prevent immune dysregulation. So in a nutshell, by various cellular and mitochondrial mechanisms, SEPSIVAC can effect a regulation of immune dysregulation and get a better Th1 response, which is similar to the tumor cell biology to kill intracellular organisms. And safety of this immunomodulator, Mycobacterium W, or what we call as Mycobacterium indicus prani, or MIP, SEPSIVAC, has shown a good radiological clearance, reduction of CRP over time. And this article was by Sehgal et al., published in 2020 May-June in Lung India, very clearly showed in this study, another study by Dr. Atul Ingale from Mumbai, along with my colleague Dr. Chandrasekhar, that the CRP levels come down by 1 dose or second dose, and even with third dose very rapidly. So a rapid normalization of CRP and reduction in inflammation, this was published in JAPI in Jan 2021, is very, very statistically significant. If you look at the improvement in IL-6, again showed the same pattern, 1 dose, 2 doses, and 3 doses would bring down the CRP, the IL-6 level also significantly, and it was statistically significant. And these two effects were shown to be associated with a rapid recovery in nearly all the patients and a quick discharge from hospital within 10 days. And this was an important study, although there was problems about the severity of illness classification, but majority of the patients admitted with COVID-19, including mild, moderate, and severe, who were hospitalized, they had an excellent response. Established safety of SEPSIVAC, because minor, barring a few self-limiting injection site reactions, there is no major adverse effect reported. And I'll just quickly show one slide of our own experience with SEPSIVAC at Nanavati. This is where I come from, Mumbai. In the last 3 months, in the waning phase of our COVID, we used SEPSIVAC for nearly 36 patients with the standard dose of 0.3 ml given over 3 doses in 3 divided doses of 0.1 ml each given intradermally. And this was for severe COVID-19 patients. I don't have time to show. All of them were in the ICU. All of them were needing high flow oxygen, and they all had underlying comorbidities. Of them, 30, that is a major numbers were males, and only 5 were females out of 36. Along with standard care, when we gave 0.3 ml of SEPSIVAC intradermal in 3 divided doses for 3 consecutive doses, on the day 14 post-treatment, we had practically 75% of them got discharged along with standard of care, and only 22% actually could not survive, or basically 6 expired, and 1 is still going on, still struggling on to the path to a slow, delayed recovery. So in summary, SEPSIVAC has diverse and potential immunomodulation properties. It can be beneficial in gram-negative sepsis and in viral sepsis, has a good clinical response for manipulation of immune dysregulation, can be added with standard of care to show significant improvement in severely ill COVID-19 patients. Prophylactic use can be beneficial for healthcare workers, and there is a very nice study called ARMY-1 which has shown that there is benefit in COVID-19 as well as reduction in the number of breakthrough infections in immunized patients. And lastly, it's a unique immunomodulator and a potential option for the prevention as well as treatment of COVID-19. Thank you all of you for a patient hearing.
Well, thank you very much again, Dr. Ansari, for wonderfully putting and correlating, SEPSIVAC in COVID-19 patients along with your personal experience. And I'm sure there are, you very rightly said, the immune dysregulation, the cytokine rush, and all those, beautiful things which we need, further explanation. But we'll come to that part of explanation, Dr. Ansari, once we go to our phase 2 of panel discussion. But by then, I will go to our next speaker, who is like you, who is one of my favorite, speaker. And while emergency critical care was in nascent stages, and while I was studying critical care, I think he was, by that time also, he was the speaker, for the emergency critical care. I'm talking about none other than but Dr. Yatin Mehta, who is our next speaker, who is an adjunct professor at NBE. He is the chairman of Medanta Institute of Critical Care and Anesthesiology, Medanta, the city. It's not a hospital, but it's called Medanta, the Medicity. He's the president of Simulation Society of Sepsis Forum of India. Our speaker, Dr. Yatin Mehta, is a teacher for Diplomat of National Board, which I passed, a long time ago, and a fellow in National Board. So, ladies and gentlemen, please welcome our next speaker, Dr. Yatin Mehta. Dr. Yatin Mehta, please, floor is all yours. Dr. Yatin Mehta, can we have you on the screen, please?
You have to let me share the slides.
Dr. Mehta, can we have your video on, please?
Can you see me now?
Unfortunately, no, but
my slides are on full screen.
Try your video and continue with your presentation.
Okay. So thank you very much for the kind
I would request technical team to look into it, but let's continue with the presentation, Dr. Mehta.
You can hear me, no? Vipin, you can hear me?
Yes, Dr. Mehta, we can hear you.
Okay, very good. So indeed a pleasure to be here, and nice to listen to the previous speakers. Okay, I bring greetings to my Filipino friends. This was when I was there a few years back in this Jose Rizal Park in Manila. It was a wonderful experience. And I bring you greetings from the hospital where I work, it's a tertiary care hospital, outskirts of New Delhi, Medanta, the Medicity. And I just want to remind my dear friend, Dr. Ansari, who spoke before us, too modest a person to show his paper. This was a joint paper review article on cytokine storm in COVID-19 in Indian Journal of Critical Care Medicine. So we have researched the whole gamut of cytokine storm and how to ameliorate it. And obviously, when you have COVID-19, you're basically there are three ways to tackle it. One is obviously you have antivirals, which will be in the first phase, then viremic phase. Then obviously prevention, you have vaccines, which will reduce the incidence or severity. And then you modulate later on in the disease because the organ failure happens, as Dr. Ansari mentioned, because organ failure happens in the later phase, you can use immunomodulators or immunostimulators. So this was a paper, randomized controlled trial of SEPSIVAC, ARMY-1 trial, Randeep Guleria from AIMS was one of the co-authors. So it was a double-blind, placebo-controlled, randomized trial in hospitalized patients with severe COVID-19 requiring oxygen. So they looked at the distribution of clinical status, and change in basically primary endpoint was the severity of the disease from category 1, which was discharge, to category 7, which was death at day 14, 21, and 28. They compared the SOFA score at 7 and 14 days. Secondary outcomes were obviously negativity and 28-day mortality and so on. And they found a significant, statistically significant reduction of severity at 14 days and at 21 days. And this paper, Dr. Ansari has already shown. This was one of the earlier papers from PGI Chandigarh. There are 4 patients you can see, and CRP reduced significantly after every dose of the SEPSIVAC, and radiologically also you can see the dramatic improvement. And this paper also from Mumbai, Dr. Ansari has shown that again, significant reduction with every dose of CRP as well as IL-6 in all the patients. And you can see another paper, the radiological resolution is pretty fast. Normally in COVID-19, you do not see improvement on day 4 or 5, but in most of these patients, it was pretty dramatic. And so was the requirement of oxygen. It was downgraded from, say, NIV or HFNC to nasal cannula or room air, and even CRP levels fell, except one patient, it fell in all the other patients. I have used SEPSIVAC in a good number of patients. I'm analyzing the data, and I'm conducting a ongoing part of ongoing PI for ongoing trial on IV SEPSIVAC in gram-negative sepsis. So I think I'm supposed to do 20 patients. I've finished 15. So these were the various trials which have been conducted and published. So this was the first one, was a 42 patients random army trial I already talked about. There's a randomized controlled, placebo-controlled trial. Second was 25 patients, effect of Mycobacterium W, that is SEPSIVAC, on IL-6 and oxygen requirement. This was hospitalized severe COVID-19 patients. This was again published. Third paper was safety of an immunomodulator. 4 patients, again published. This was in severe patients. So it has been used from mild to prophylaxis in some, to asymptomatic patients to severe patients. So all categories, severity of the disease, it has been used. Then you have early experience of high-dose IV SEPSIVAC. Now this is coming to SEPSIVAC IV, because SEPSIVAC, one of the questions was local irritation because of sub-Q injections, and particularly patients who are not very sick, giving 3 sub-Q injections at 3 different sites can be a little of have a bit of discomfort. So IV SEPSIVAC is being used now. So 6 patients, severely ill COVID-19 patients, again published in significant benefit. Then there was a 50 patient study, effect of SEPSIVAC on lab and radiological improvement. These were hospitalized RT-PCR confirmed COVID-19 patients. There was 15 patients in clinical improvement with immunomodulator in severely ill COVID-19. 15 patients. This was severe COVID-19 infected patients. So as I said, it has been used for mild patients to moderate patients to severe patients. And this last one is still undergoing analysis, clinical trial of SEPSIVAC in critically ill COVID-19. So this is critically ill patients, 40 patients with severe, critically severe disease. So this is an ongoing study. And then you have SEPSIVAC in treatment of an observational study, good number of patients, 117 patients of moderate COVID-19 disease. Then a randomized double-blind trial to compare the efficacy and safety of SEPSIVAC. This was a large study, 480 COVID-positive hospitalized patients, but not critically ill. So they were mild COVID-19 disease patients. And then this was a voluntary sort of study, 3,800 patients, I think, which was earlier presented, to look at the innate immune modulation and resistance to development of COVID-19, which was also a significant positive study in large number of volunteers, employees. And then you have this 96 subjects of healthcare workers, again looking at the innate immune response. This is a prospective study and high-risk healthcare workers, so significant. Now this was publicized quite quite a lot in the media, print as well as visual media. And CSIR, which is the highest authority of Center for Scientific and Industrial Research in India, with the help of which the studies were conducted, and it has now been approved in India by Drug Controller General of India for emergency use in COVID-19. So thank you very much for your attention.
Thank you very much, Dr. Mehta, for wonderfully, showing the evidence-based medicine. And I'm sure you have also come across, a lot of patients who have been treated with SEPSIVAC during these COVID times. We'll come to that part, Dr. Yatin Mehta, once we have or start our panel discussion. But by then, let me go to our next section, ladies and gentlemen, and that section is, panel discussion. So while we go to that section of panel discussion, let me introduce our panelist, next panelist who is going to be part of that panel discussion is Dr. Rajat Agarwal. Dr. Rajat Agarwal, he is an additional director and head of the Department of Critical Care Medicine at Fortis Escorts Heart Institute, New Delhi. He is a teacher of critical care medicine in Diplomat of National Board, what we call is DNB, and an Indian Society of Critical Care Medicine. May I have Dr. Rajat Agarwal. Dr. Rajat Agarwal, welcome to this. Good evening.
Good evening. Thank you. Thank you, Dr. Vipin.
Yeah, Dr. Agarwal, I would like to have your opinion, because while we were going through the second wave of COVID in India, Delhi, Mumbai, along with other cities, but mainly it was Delhi and Mumbai who were going through, various COVID patients and a lot of problems out of this. What is your experience? How do you relate, the SEPSIVAC, how it was beneficial, and what all we have faced during that period of second wave of COVID-19 in India?
So, thank you, Dr. Vipin. So, after a very detailed and nice presentation by our previous speakers, Dr. Mehta sir and Dr. Ansari. So, as my fellow panelists will agree that all intensivists in India and all over the world face the wrath of COVID-19, especially during the April and May. So 2 months were really bad for all of us. We saw a lot of mortality and things were quite serious. But yes, we started using SEPSIVAC in the month of April. To begin with, I was also a little hesitant, I will admit, and we started in using, as the dose mentioned, intradermally, 0.3 ml in 3 different sites. But because it was difficult in the peak of pandemic to monitor the patients, to monitor their efficacy, we got some research and some paper which showed the evidence that intravenous use can also be there for SEPSIVAC. So we then started using in patients of gram-negative sepsis post-COVID. I'm sure that every intensivist knows that there was a high incidence of gram-negative sepsis post-COVID, and everybody was treating that. And so we switched to intravenous route, 0.3 ml intravenously once a day for 3 days. And we use in post-COVID complications in gram-negative sepsis. We started a small trial for that, and currently we are just enrolled 25 patients. Out of that, obviously we are just analyzing our data. But whatever raw data available with us and whatever clinically what we saw was that yes, the number was too small, I admit, to show any significant difference in mortality. For that we need a large sample size. But the difference in Apache 2 scores, the SOFA scores, the ventilatory requirement and requirement of vasopressors in gram-negative septic shock was significant after the use of SEPSIVAC. So we could demonstrate that yes, patients presenting with high SOFA scores of more than 15, 16 with multi-organ failure, the ventilatory requirement and especially the lactate clearance and vasopressor requirement decreased significantly after the use of intravenous SEPSIVAC for 3 days. Though still we need to have larger study to know the efficacy, but whatever experience we had and whatever research we did has shown these clinical improvements significantly. Over to you, Dr. Vipin.
Dr. Vipin, please turn on your microphone.
Dr. Vipin, I cannot hear you. Sorry.
Dr. Rajat, can you hear me?
Yeah, I can hear you now. Yeah.
Yeah, all right. So I'm going to my next panelist now, and our next panelist is Dr. Shweta Annankhere. Dr. Shweta, can you hear me?
Yeah, I can hear you.
Wonderful. Dr. Shweta is a senior critical care consultant at Care Hospital, Banjara Hills, Hyderabad. Hyderabad is a wonderful place. She has authored many research published articles published in renowned journals. So, Dr. Shweta, like yesterday we were talking about the complications because India have passed through the second wave, and now we are talking about third wave. Philippines is going through right now a very crucial time of COVID waves and all that. However, we have passed through and now we have moved to a step where we are coming across with a lot of complications. Like one of the complications yesterday we discussed about cellulitis. However, a lot of people are coming with dengue, chikungunya, pains, cough without any unknown etiology. How would you like to describe this? And how is your experience, post SEPSIVAC, whether these complications been reduced or how was it? Dr. Shweta, please.
Yeah, thank you, Dr. Vipin. Thank you for inviting me for sharing my experience. First of all, I just want to share my experience of using SEPSIVAC. So in the first COVID wave, when there was dearth of information about a lot of drugs and what actually to use, I just came across the articles on SEPSIVAC and wanted to use in my ICU. So I was at that point of time working in an ICU in which all the patients were on ventilator and were having CT severity score more than like 22/25. Actually, I wanted to use SEPSIVAC in patients who are on high flow oxygen who may require ventilator to see whether we can prevent the ventilation. But initiation of SEPSIVAC was one problem because it contains Mycobacterium W. So my colleagues who were working in that ICU were like skeptical. So I had to tell them it's heat-killed Mycobacterium, which is non-pathological. And the other problem they heard was that it may form some ulcer. So what I did, I started using it in my ICU on the ventilator patients. And I used to give the medication myself. So 0.3 in 3 divided doses, that is 0.1, 0.1, 0.1, once a day for 3 days. And I saw good effect. So that the X-ray clearance was seen over a period of 6 days. We could see IL-6 level decrease over a period of 6 to 8 days. Regarding the mortality, if we say, it is very difficult to tell because I have used it in 15 patients. Again, it is a small number. And mortality is not only defined by one drug. There are many factors which may define the mortality, like the severity of the illness, the age, the comorbidity of the patient, and the patient, when does he arrive at the hospital. Now during pandemic, the resources were so low and the bed availability in the hospital was also very difficult. So many patients used to come to us at very late stage of the disease when they were quite sick and having multi-organ dysfunction. So mortality, difficult to say at that point of time, but yeah, we could see no adverse effect, and that made my colleagues to use it in their ICU. So when it was used in the ICU where the patients are on high flow oxygen or NIV who are not on ventilator, we could see that 60 to 70% patients did not require the ventilator. The oxygen requirement decreased over a period of 6 days. The respiratory rate also decreased. And again, we did not see any complication. We did not even see ulcer because the doctor themselves used to administer the medicine. Now, regarding the second part, like the post-COVID complications. So we have not seen any complication with the drug, but post-COVID patients now, after the regression of the second wave, now we are getting patients who are developing cellulitis of the lower limb. We know that thrombotic complications are common in COVID, but these patients, we have screened Dopplers and all, there is no DVT, but cellulitis. Now, cellulitis is a gram positive infection. So we don't know whether this cellulitis lot of cellulitis case, are they a complication of COVID? It is difficult to say.
Dr. Shweta, you're coming across a lot of complications.
Yeah.
COVID with so many patients. Thank you very much for this insight. Dr. Shweta, we'll have a panel discussion, but before we come with all the panels, let me go to our next panelist, who is a very senior doctor from Philippines. So, ladies and gentlemen, we have Dr. Hubert Benedicto, who is an associate professor at the University of Philippines College of Medicine. He is the chair for critical care unit management team and the vice chair for patient services at the Department of Medicine at UP Philippine General Hospital. He is likewise the head of public health and domiciliary unit at the lung center at the Phils. Let's welcome Dr. Zubert Benedicto. Dr. Zubert Benedicto, very warm welcome to you to this webinar. How are you, sir?
Thank you very much for that kind introduction. And I would like, of course, to thank everyone for inviting me. This has been a very good webinar. I've learned a lot. And thank you very much, of course, for the speakers and panelists for sharing their experiences. So basically, I'm really intrigued because I feel that this really is an innovative drug because it hinges on what I term as the Goldilocks phenomenon. You know, you try to immunomodulate the immune system, not too much, not too little. It tends to dampen certain processes, but it tends to enhance certain processes as well. So the just right or the Goldilocks phenomenon. And I'm really intrigued and interested with that concept because as what was shown, in certain things you need to be to have an agonistic effect. For certain things, you have to be in the antagonistic side in order, let's say, for you to derive the maximum benefit. And I think that was shown on a lot of studies. But moving forward, of course, since we're going to have a panel discussion later, I would really like to see if what is the optimal timing of giving the drug itself. I mean, let's say, for example, for COVID, will it be optimal once you see the patient as RT-PCR positive or COVID confirmed? Because as what was shown in the diagram earlier, of course, we would not like the patient going into the third stage already. And definitely, maybe, you know, by enhancing or immunomodulating the innate activity of the immune system at that particular point in time may be beneficial. Secondly, of course, since I'm from the Philippines, we do know that a lot of us tends to practice, you know, by anchoring our practice in guidelines. So we do know that we have a sepsis guideline, and it's not just the global guideline, both for sepsis and for COVID-19. But of course, we tend to have an adaptation to it. We have a Philippine guideline both for sepsis and COVID-19. And principally, we do know that for sepsis, there's that bundle of care approach as well. So maybe I'm just thinking at the top of my head, considering the benefits that I've seen from the literature that was presented, this should have been a part of the bundle of care. I mean, especially the first R of the bundle of care. Once you diagnose a patient who have sepsis, maybe you can derive a lot of things once it's there. Same goes for COVID-19, of course. We do know that there's, you know, a lot of literature that comes out. We are deluged with a lot of literature right now. And we do know that, of course, not all the drugs, you know, based on subsequent use will last the test of time. Some of them fall off from the guidelines. Some of them needs to be bolstered in the guidelines, or some of them needs to be, of course, niche in the guidelines. What is the proper positioning, etc. So I do believe that this drug is resting on a very innovative yet logical framework or theory. And maybe it's about time that we see, as what was shared by the panelists, you know, trying to stretch the come-ons and attributes of this drug. Since we are all clinicians, we are all frontliners, we would like to take off from what the drug is trying to offer us and maybe test it really amongst our patients and to possibly learn as we go through with this pandemic. Thank you again for the opportunity, sir.
Thank you very much, Dr. Benedicto. All your points and questions have been noted, or maybe once we start the panel, discussion. So your one question I'm going to take first, which is, the timing. So we'll start by then, but just to complete my panel here, I would like to invite our next panelist here, Mr. Mahidwa Sisodia, who is the head of international division at Cadila Pharmaceutical. Mr. Mahidwa Sisodia has a good and rich 5+ years of experience managing international business for various companies. And with Cadila, he has been associated for last 6+ years. Mr. Mahidwa Sisodia, very welcome to you. A very good evening to you once again. How are you, sir?
Good evening. Thanks, Vipin. Good evening to all of you. And before I say something, let me thank esteemed speakers, panelists, all doctors, not only from Philippines, but I know they have joined from other countries also. And also to Biocare team for putting up this webinar. This has been really interesting. And yeah, go ahead.
So, Mr. Sisodia, before we, go to and have entire panel in place, just few points, maybe, some of them across the globe would like to know what is our strategy of international expansion and the stock availability? Because as we understand, COVID comes suddenly, and then, like we saw, there was oxygen requirement, and then there was, a lot of mismatch in the drugs people wanted, but it was not available. SEPSIVAC is one such drug, which availability is very, very important. And, internationally, in how many countries we are expanding and how we are going about it. Would you like to have throw some light on this, Mr. Sodia?
Sure. Thank you once again. So we have mentioned during the presentation of Cadila Pharmaceuticals that we are present in more than 100 countries. And when we say these 100 countries, they are like our homes outside the home. And we have been always well connected with the situation there. And what we have seen in recent days basically triggered us and rekindled the memories of period during April and May in India when we had a tough situation. And we could see the similar situation rising in many of the countries in last days. Our heart was with them, and we thought that how can we come into this situation and help them. So we decided to approach regulatory authorities of this country, and I must tell you that we got very positive response from all the regulatory authorities. Also, we were supported by the embassies of these countries in India and Indian embassies in this country. So what we are looking at is to get permission, special permission to import and distribute these products. And I'm hoping that within few days we should have this, which will be helping us to make the stocks available, which again is not a big issue because we have decided to keep the stocks ready for this kind of product considering the situation. So within few days we should be up and running. And also I must tell you that what we have decided during this period is to work with the companies like Biocare in Philippines and other companies in other countries who have been specializing and bringing a wide range of product portfolio for COVID. So this is what I can talk about our plans as far as the SEPSIVAC is concerned in these countries. And we are well established in many of these countries through our own infrastructure or the infrastructure of our partner, not only to market, but to distribute as well in very efficient way.
Thank you very much, Mr. Mahidwa, for giving a comfort and telling that you tell us and SEPSIVAC will be made available in your country. Thank you very much. I think that will give a lot of positivity to all the medical fraternity and our colleagues in Biocare sitting there that immediately the availability is not an issue. And we are going to various countries. Thank you once again, Mr. Sisodia. And may I now, so with this, we complete our panel. So may I request all our panelists to please, switch on their cameras. Dr. Yatin, Dr. Ansari, Dr. Rajat, Dr. Shweta. Can we all line, please, so that we start our panel discussion? Dr. Benedict, please. Wonderful to see everybody. Dr. Shweta, I think Dr. Kumar is also there. So Dr. Shweta, we can't see you, but definitely we can hear you most likely. So
yeah.
Thank you very much. Thank you very much for all coming online. Now, I would like to start this panel discussion with, I understand Philippines is going through a tough situation, Dr. Benedict, and what I see the data, there are a lot of cases and only 10 to 12% vaccination is happening. And we understand, it's not going to go away, the herd immunity, that 70% of population, unless they are there. So SEPSIVAC is one of the very good drug. Dr. Shweta, good, we can see you now. Thank you very much. So I start with the question from our Professor Benedict from Philippines. As I think this question I would give it to all the panelists. What is the right time? What is the right day to start SEPSIVAC for a patient? And if you can divide, mild, moderate, critical. We can start with Dr. Kamar and then we can go to Dr. Yatin and Dr. Ansari for this. What is the right time or day to start to COVID patients?
Okay, so let me clear, I am not a clinician treating sepsis or a COVID, but the science which I know and immunity or immunology which I know tells me that to boost innate immunity, all time are good. So the moment you see the patient first, you can administer the drug without waiting for him to deteriorate or to get a right time, because it does not do any harm. It only enhances body's reaction to the organism. And as Professor Benedicto said, it does two jobs. It increases body's mechanism to fight against respiratory viruses by having a potent action on TLR2 and NOD2, which are responsible for killing intracellular organisms. But at the same time, the deleterious effect of the infection comes from TLR4, 7, 5, 9 overexpression. So it reduces those, or it suppresses those overexpression. And then the other effect which we see in gram-negative sepsis and COVID as subsequent to TLR overexpression. So it works like a double-edged sword, but a friendlier sword. It cuts the enemy on both the sides.
Dr. Yatin, would you like to add something?
Yeah, see, now the initial phase of, as Dr. Ansari mentioned, is a viremic phase. More the virus, higher the viral load, more likely the severity of the disease. So if you can attenuate viremia, you can reduce the viral load, you may be able to make it milder, the disease. So I think if you give it in the viremic phase, initial phase also, as Dr. Khamar rightly pointed it out, it will reduce viral load and it will reduce the severity. Several studies have also shown in patients who are exposed high-risk cases in healthcare workers and so on. Now the second, as Dr. Ansari again mentioned in his review article on cytokine storm, that in the week or 10 days after the disease onset, you get this cytokine storm. Of the cytokine storm, because the cytokine storm, there is also macrophage activation syndrome, and there is high levels of cytokines, which are again not only they lead to multi-organ failure, but they also determine the severity of the illness. Higher the IL-6 and TNF alpha and so on, IL-1, pro-inflammatory cytokines, higher the severity, more the severity of the disease. So if you are able to immunomodulate at that stage, that is where toci and all were used, I think steroids and toci. This would add to the armamentarium and this would attenuate the cytokine storm, which may reverse the progression to multi-organ failure. So at that stage also it may be beneficial. The side effects, as rightly pointed out, hardly any side effects, local pain on injection. So I think it is a good addition to what we have.
Thank you very much, Dr. Mehta. Dr. Ansari, you have shared one, a series about 36 patients you have treated who were very seriously ill in ICU. What is your experience on what day you started using SEPSIVAC and so that it gives a clear picture to Dr. Benedict, when it's to use in these critical care patients?
So, as Dr. Yatin sir had very clearly, defined and mentioned, I think a very important fact to understand is that you have time to modulate the immunity before it has gone for a toss. When things have gone out of hand, the repair process is always difficult. At the same time, we have to understand that viral sepsis is little different than bacterial sepsis, because you can always see that there is leukopenia and there is absolute lymphocyte deficiency, in which situation your adaptive immunity is going to be lagging behind. It's always your innate immunity which is be the leading edge, and if you can strengthen it, as Dr. Kamar also said, and it is very interesting if you go through the mechanism, the four important things that it does: the TLR2 agonism, the NOD2 agonism, by which your host effector cells are trying to identify the virus and releasing favorable cytokines, especially the interferons. If it is timely done, that can ride on your adaptive immunity also very well. So your innate immunity, timely intervention with SEPSIVAC can boost your adaptive immunity to catch up with the disease. And very importantly, it has shown to have P38 down regulation, which is important mitogen activated protein kinase, a very important signaling pathway by which your cells get activated and your T cells are coming back into good action. So I think no doubt about that. You can't give it to everyone unless you are doing a prophylactic study. But if any person who comes with moderate illness, right in the beginning, when there is an increasing oxygen requirement, in conjunction with an antiviral, is the best therapy to be started. And you may need to give some steroid as another add-on adjunct immunomodulator. In that case, as Dr. Yatindra very clearly said, we may be able to avoid more what is called toxic drugs like tocilizumab or Janus kinase activator inhibitors.
Thank you, Dr. Ansari. Dr. Ansari, you said, but before I go to the next question, may I, Dr. Benedict, are we okay? Have you got the response to your question, Professor Benedict?
Yeah, I think they answered my query well. And I think that's the good positioning as well of SEPSIVAC, that once you identify, let's say, the patient to be afflicted either with sepsis or COVID-19, then you very well can give it, bolstering their innate immune system.
Professor Benedict, I understand and assume, there are many combinations which are being used in Philippines also, like other countries. So if I ask my panel, I mean, anybody can respond on this. How about drug interaction when it comes to SEPSIVAC, or how this is different with comparing to other immunomodulator? Or maybe if I have to be little specific, how about tocilizumab? So how do we compare, and where SEPSIVAC has got little more advantage? Some have experience, some will have R&D data. So again, maybe if I start with Dr. please, and then Dr. Mehta.
So sorry, I will be the one answering first. Yes, okay. So right now, what we're following is our COVID-19 living documents. Basically, what it contains is that it's a rapid review of sort done by some epidemiologists in the country, and of course, as well as clinicians and frontliners. So bottom line, once the patient is on oxygen, we are giving that patient your dexamethasone, and subsequently remdesivir can also be given. We usually give remdesivir for 5 days, and then subsequently for tocilizumab, to answer your query, tocilizumab is more reserved actually for patients who might be having an increasing oxygen demand. So in terms of their oxygen support, it's increasing, and you see that, let's say, the inflammatory markers are likewise increasing. So it's that particular group that may be administered with tocilizumab. So that's the way right now. But I do know that, of course, in the country right now, in Philippines, there's relatively a shortage of tocilizumab. So basically, it's really just dexamethasone for now, and then subsequently remdesivir. Of course, we still go with some other treatment like your low molecular weight heparin, etc. So it's still basically anchored on the severity of the patient, and then as what I've mentioned, it's basically founded as well in our COVID-19 living document.
Okay. Now, Vipin, since you asked me, and I have to leave in another 5 minutes.
Yes, Dr. Mehta, please.
Another talk, yeah. So I was one of the co-authors of a randomized controlled large multi-center trial published in Lancet on tocilizumab. Okay, it was moderate to severe cases. It was randomized blinded study. And we found really it did not meet the primary endpoints. I'm not, although on subgroup analysis, we did find some benefit in severe cases, but otherwise we did not meet the primary endpoints. It was a negative study. I am not saying that it's not a good drug or it should not be used. What I'm trying to say is tocilizumab was basically being used in moderate to severe cases when the patient's either symptomatically is getting worse, that means the oxygen requirement is going up, or is radiologically is getting worse, or the other parameters get worse, or if you have increasing CRP or increasing preferably IL-6, because IL-6 receptor blocker. So if you have demonstrable these criteria or clinical criteria of CRS, then TOSI is used. But TOSI's main problem was with TOSI was it is a severe immunosuppressant. So the incidence of secondary sepsis, bacterial as well as fungal, was very high in these patients. And that we found out of this study also, most of us who have used it during the second wave, we found a significant increase. That's why India saw such a huge incidence of mucor also, because we had a high incidence of secondary sepsis, which killed the patient. The patient was not going to die of COVID-19, he was dying of secondary sepsis, whatever, bacterial or fungal. So that was the main problem with TOSI. And also TOSI is an expensive drug. And there was a shortage in India, as like in Philippines, Dr. Benedicto mentioned it to us. So it has to be used with a lot of caution. Side effects are significant. And there are specific indications when you should use it. With SEPSIVAC, the advantage is, A, it is very safe, and B, it is not that expensive. So really, you're not going to harm the patient, if at all. I mean, otherwise, TOSI can harm the patient.
Thank you, Dr. Mehta. Dr. Kumar, would you like to add something to it? Wherein, I clearly see TOSU is immunomodulator as well as suppressant. So Dr. Kumar.
Okay, so generally, almost all immunomodulator induces immunosuppression, which includes tocilizumab, which includes steroids. So they are all known to induce immunosuppression because they work only on one side. They suppress. Whatever is upregulated, they will downregulate it. They don't see what is right and what is wrong. The advantage of SEPSIVAC is it tries to re-establish immune homeostasis. That means whatever is down-regulated, it gets up-regulated, and whatever is up-regulated, it gets down-regulated. So at the end, if you use an adequate amount of SEPSIVAC in each and every patient, you are not likely to see immunosuppression when they recover. So typically, gram-negative sepsis or COVID-19 recovery associated with immunosuppression. While when you use SEPSIVAC, that component will be minimal or not there. It depends on the dose and the severity of the disease and all other factors, but it mainly tries to re-establish immunohomeostasis. Other part is tocilizumab and steroids and other immunomodulators, they work directly on the cytokines and other things. While SEPSIVAC does not have a direct effect on those cytokines, it works through body's own immune system. So it stimulates body's system in such a way that the genesis of this cytokine is altered. So it's body's own defense. It takes little time compared to immediate effect which you see with the immunosuppressant, because it goes through the body's immune mechanism. But then it works through innate immune mechanism, so it does not take days. It takes few minutes to few hours to manifest its action.
Thanks. I think Professor Benedicto, so this is, while we are using TOSU and SEPSIVAC really have an advantage, and I'm sure others panelists also have same type of experience. And I see Dr. Shweta, nodding her head, and I would, Shweta, I come to you, but before that, let me just tell our audience that Dr. Mehta had to go for another webinar, so we are sorry, and we thank him for joining, but he left. Maybe all the questions to Dr. Mehta you can please put on your chat, and we will revert to you after talking to Dr. Mehta on this. So coming back to our panelist again, Dr. Shweta, I see you're nodding your head and you want to say something, please.
Yes, definitely. So as Dr. Mehta already said, tocilizumab is a immunosuppressant, so it may decrease the immunity. So before we give tocilizumab, we have to make sure the patient does not have any infection. So if the patient has infection, it may worsen. But when we are using SEPSIVAC, we need not have that problem in mind, because SEPSIVAC is being used for even gram-negative sepsis. So maybe in future, we may see that when we are using SEPSIVAC in patients with COVID, it may even prevent the gram-negative infections. Right now, we are doing a study of treatment of the gram-negative infection as an immunomodulator. Maybe future studies may show that. So we need not worry of the sepsis in the patient, secondary bacterial sepsis in COVID patients when we are using SEPSIVAC.
Thank you, Dr. Shweta. Dr. Ansari and Dr. Rajat, we have a question. I'm not sure what is your experience about this. This is about prophylaxis. I understand we have seen one study, but have this been used as a prophylaxis to some of your patients, and where you have some experience, user experience, may not be having a data, but I understand Dr. Kumar can again come later in this. But first I go to Dr. Rajat. Any of your experience on this would you like to add? And then I go to Dr. Ansari for this.
So for prophylaxis, yes, there has been now a lot of data is coming up, and as discussed by one of the speakers in the panel, that it has shown significant efficacy for prevention of COVID-19. I think Dr. Khamar presented that data, and the efficacy was more than 93%, which was quite highly significant. I personally don't have that experience of using SEPSIVAC as a prophylactic for prevention of COVID-19, because we usually are dealing with intensive care when the patients are already lined up with severe COVID-19 infections. So Dr. Ansari might be having some experience with that, but yes, the literature which has been presented so far sounds very promising. And if the government guidelines can put it in as a part of the protocol for a larger scale studies, I think it's worthwhile doing the study on prophylaxis for prevention of COVID-19 with SEPSIVAC. Thank you.
Dr. Ansari, over to you.
So I completely agree with Dr. Rajat that we don't have enough opportunity to use it in the prophylactic capacity. But this study which was done was very interesting, because I went into the specifics, and this study was prophylaxis of COVID-19. There were 32 patients, 32 healthcare workers working in bone marrow transplant unit, and they were compared with another 14 or 15 control patients, and equal number, and 3% versus 46% of breakthrough infections over a period of 3 or 6 months. Very interesting actually, and it cannot be just a statistical kind of change. So I think it really needs to be looked at for high risk patients, like we are talking about monoclonal antibody therapy, cocktail therapy. Probably for high risk asymptomatic people, giving it very early could be a way to look at how things evolve before even they get symptomatic. Maybe that is something which study should be done, just like a comparator to monoclonal antibody cocktail, as we all are using for high risk patients. That could be one interesting way to look at it. And apart from that, all those patients who have already had vaccinated, whether in that population using it to prevent Delta plus strain kind of infection with whole genome sequencing would be worthwhile. So I think these should be areas of future research.
Dr. Kumar, would you like to react to what, 2 pointers Dr. Ansari mentioned? One is we have study, and other is, about the vaccination and all.
Okay. So, I mean, this is kind of a disruptive science. See, all of us are trained right from the second MBBS onwards that we need a humoral immune response to get ourselves protected from the infections. Correct? I mean, everybody has taught us and we have learned that all throughout and we remember that by heart. And we always look at what is the titer. And we also know that titer over time comes down. Correct? So now, I mean, if you have look at now recently what the American data has come, the Pfizer vaccine, in some pockets, the efficacy has come down to less than 50%, which was supposed to be the best working vaccine, having 90% plus efficacy. Against Delta, the efficacy for Pfizer and Moderna has come down to around 50%. And that's the one of the CDC data which has recently been published. The reason it happens is that antibodies, once produced, it decays over time. And if you compare that with other vaccines, say for example, hepatitis B. Hepatitis B, when you give vaccine, your protective titer and the achievable titer, the gap is very wide. It's more than 1,000-fold that you achieve by vaccine, and that's why you remain protected for a longer time. But with all the available vaccines which are currently approved around the world, the protective efficacy titer, which is identified by WHO and CDC and other groups, and the highest titer which is achieved by the available approved vaccine, the ratio is anywhere between 2.5 to 10-fold higher at a peak. And then it drops over a time. And then unfortunate part is Delta came in those populations at a fag end of their titer after 6 months of receiving vaccine. And that's probably the reason it is gone. Now, when you come back to the innate immune response, SEPSIVAC activates macrophage and NK cells that are responsible for killing of the virus as it enters the body. And its mechanism is different. I mean, if you look at immune escape mechanism of COVID, immune escape is because of the IFN alpha, type 1 and type 3 interferon, while SEPSIVAC works through type 2, which is IFN gamma. And IFN gamma is hallmark. And what we have observed is, unlike vaccine, you have to give 2 doses and wait for 14 to 28 days for the full benefit to achieve. This benefit starts immediately within 2 hours. And in whatever experience we have, it has been sustained for more than 6 months. And then, I mean, which is not published, but in a group of people, healthcare workers who are vaccinated. I mean, in India, we know that even the healthcare worker, infection rate was very high, and we lost some of our colleagues who were frontline workers or treating doctors. So in those groups, there's a limited data, but in those groups, the infection rate, breakthrough infection, was significantly higher. But then few people tried to receive SEPSIVAC after 2 doses of vaccine, and the breakthrough infection was significantly low. And by one statistic, it was 16-fold lower, 16-fold lower if you have received 2 dose vaccine with SEPSIVAC versus 2 dose vaccine alone. And vaccine which was used in India was primarily Covishield in that group. So, I mean, and then because of the mechanism of action, there is no contraindication of using 2 together in a sequential manner. So if you have received Covishield or Covaxin or BioNTech or any vaccine, you are eligible to receive it. The other part is that SEPSIVAC by itself works as a vaccine adjuvant. So it is likely to boost your antibody titer further rather than reducing it. So there is no contraindication of using it. The only request, because we don't have a lot of data, give adequate spacing between, if you try to use SEPSIVAC following your vaccination or preceding vaccination, give adequate time between the dosage of the 2. Like current recommendation is between 2 dosage of vaccine, any vaccine, you have to have minimum 28 days. So at least give that much time before you start using any of those 2 modalities, whether vaccine or non-specific modality like SEPSIVAC. And that should help you protecting the frontline workers who are more vulnerable better.
Thanks, Dr. Kumar. So I understand, you know, you said a couple of things, which is it is at least it is effective for 6 months. What we look here is memory cells are hardly for 90 to 120 days. It's not much more than that. So 6 months looks pretty good. Still, it looks pretty good. Second is, vaccine now again. In fact, Pfizer and all in US, they started saying there is a booster dose after 8 months, right? So after these 2 doses of vaccines, we can have SEPSIVAC. Yes, Dr. Kumar, you want to say something?
So the point which I did not mention, and I remembered that when you said that Pfizer, etc. See, all the vaccines are specific for a particular antigen. So when the virus mutates, the antigen profile changes and vaccine efficacy automatically drops or changes to the different level. And that's the reason for an influenza virus which is highly mutating, you have to have a new vaccine every year. You don't establish permanent immunity against influenza virus because the virus mutates, and then the vaccine which comes into the market takes care of those novel antigens and you get protected for that season. And the same thing has happened for COVID-19 or SARS-CoV-2 virus, because virus mutated so much between the first Wuhan strain to the Delta strain that the efficacy, even when you are, immunized recently, the 4.5-fold decrease in efficacy against Delta virus compared to the Wuhan virus with all the vaccines, where efficacy was maximum with Pfizer and Moderna vaccine. Even there's a 4.5-fold reduction in antibody titers against this. Effective neutralizing antibody titers, there's a reduction by 4.5-fold. And there's a nice, beautiful article in Nature Biotechnology to that effect. And then there are more when you go to the WHO and listen to the people and the talks. This is more and more people getting, re-evaluating and reconfirming this finding. The beauty of SEPSIVAC is it is independent of antigen. It does not recognize antigen per se. It does not recognize SARS-CoV-2 Wuhan strain or SARS-CoV-2 Delta strain. It just says it is a virus which needs to be identified because it has a particular pattern which is conserved. As Dr. Ansari said in the beginning, it recognizes conserved patterns, and conserved patterns are specific to the virus. They don't change. They are evolutionary conserved. And if you go back and trace back, evolutionary immunology, innate immunity was the first immunity or first immune response to protect organism, and that has been conserved over generations and over evolution. So it is fast, efficient response, and it recognizes only the pattern and not the antigen, not a specific protein or a carbohydrate. And so it works equally well against the mutants.
So we know COVID is not going to go away. It is going to come back again, and more we study and more we come to know about this. So Professor Benedict, as Dr. Kumar very rightly said, role everywhere. One of your questions, where and when to use, and in what sort of indication. Prophylaxis, we have a study about 4,000 patients. Now patients who are vaccinated, because in Philippines it's only about 12% population which is vaccinated. But even though they are vaccinated, and if we give a dose of SEPSIVAC, wherein study is going to publish hopefully very soon, it is going to give a 14 to 16-fold more protection, right? Otherwise the memory cells are only for 90 to 120 days, and at the most vaccine is gonna give you a protection for 6 months. You add this, the longevity which goes further on this. But while we are talking about all this, I think I have one question, which is, what next? Every time it comes, what next? And this question from my side goes to Mr. Mahidhar Sisodia. Mr. Mahidhar Sisodia, what is the plan? What is the game plan now? What next? Where do we go from here?
No, so every day when we are getting update and more information, and especially from Southeast Asian countries, as I told you in the beginning, our hearts go out to everybody out there. And what we are looking at is that how do we communicate more and more? Like we have the communication like what we are having today, so that there can be more information be made available to all the stakeholders there in those countries. At the same time, thinking about the ways to make the stocks and the product available in the country. So with that, I still remember the stories during the month of April and May from India, when we used to talk to our Indian operations colleague, and they always used to come and used to share the stories of SEPSIVAC, how it has helped the medical fraternity at large to overcome those challenges. We are looking forward to play the same role during this period, knowing well that situation is getting difficult and it's time to act now. We are really looking forward that we will be able to secure some special permission in these countries and play our bit through our partners like Biocare in these countries and help humanity at large and overcome these challenges. So that is what we are looking forward, and hopefully within few days time we should be up and running in these countries. Thank you.
Thank you very much, Mr. Sisodia. I have a few more questions, but before I take, it's just for all the audiences, if you want to ask any question to any of our panelists, you have, a question box you will find on the control panel, in the course of during any time, if you want to put in a question, please put in the chat box. It will come to me, and then we will have, our panelists to respond to those questions on this. Right, so you can please have your question while you are putting your questions in that. I have to all our panelists, I have one question which is coming to me. It says, what laboratory procedures to request to determine if SEPSIVAC is working, and what to do we expect to see in the results. I think this question is not only for SEPSIVAC, it's for any medication. While we give any medication, what sort of laboratory procedures or requests to determine whether it's working or not working. May I start, some response from Dr. Shweta? You are nodding, so would you like to respond first on this, and then I go to other panelists?
Yeah. So I think we should not only see some biomarkers, we should also see the clinical condition of the patient. So in clinical condition, whether the oxygen whether requirement of the oxygen is decreasing, whether the respiratory rate is decreasing, whether the patient actually is requiring ventilator or not. Then when we come to the imaging, whether there is improvement in the chest x-rays, that's what we have to see. And then next is the biomarker, whether there is decrease in the IL-6 levels. That's what. So we have to combine the whole data, and more importantly is the clinical data.
Okay. Dr. Rajat, any view on this?
Yeah, so Dr. Shweta has rightly pointed out that the patients in which we used in our hospital was predominantly gram-negative sepsis and post-COVID gram-negative sepsis complications. And we noted down the various parameters like SOFA scores, requirement of oxygenation PF ratios, and requirement of vasopressors. So in these three parameters, we noted significant improvement with the use of SEPSIVAC over the period of time. Mortality benefit, yes, we could not ascertain because our sample size was not powered enough for that, and we are still doing the analysis of our patients' data. But yes, these were the main clinical parameters which we found to be significantly low after the use of SEPSIVAC in patients of gram-negative sepsis.
Thank you very much. And before I go to Dr. Ansari and Dr. Kumar, Dr. Benedicto, how are you managing? You may be given other drugs you are giving, and how do you, and what sort of tests or parameters you look for while a patient, COVID patient, comes to you and you put them on various drugs? What sort of parameters or tests you generally go for in Philippines?
Yeah, so just like what was mentioned, so basically we're going on some integrative approach. We integrate everything. So basically we look at inflammatory markers as well, your CRP, your ferritin, and then subsequent LDH, and then subsequently, of course, the oxygen requirement as well of the patient. And then we just monitor it regularly. We monitor those parameters regularly, integrating it with the patient's clinical scenario. Then we go on from there if the patient is really responding to the treatment.
Thank you, Dr. Benedicto. Before I go to Dr. Kumar, Dr. Ansari, how's your experience? What, some important parameters you look for? And especially in, the study you shown in 36 patients, and, what sort of parameters you did watch for?
So I think Dr. Shweta, Dr. Rajat both had very nicely kind of elaborated, and Dr. Benedicto very nicely said that, there is a COVID gram risk score, and there is also something called call score. But in general, you cannot, comorbidities are the same. Age remains the same. So LDH and absolute lymphocyte count, these are two very important, readily observable factors. which will try to give you the pattern. On top of it, if you had a very high D-dimer level, that as a falling trend. But I think of all of them, CRP, absolute leukocyte count, LD, and IL-6 is an expensive test, may not be available everywhere, but at least every 72 hours, looking at the IL-6 if it was high would be an important trending device. And finally, a SOFA score for a person who was severely ill will give you an idea about the PF ratio, the neurology, the kidney function, the liver enzyme. So it gives you a snapshot of all the organs. Biochemically, that's one. And clinically, if the patient's respiratory rate is coming down, as Dr. Shweta said, and the PF ratio is improving, I think that's one of the most healthy indicators of an improving clinical condition.
Thank you very much, Dr. Ansari. Dr. Kamar, this is Dr. Kamar's baby actually, the SEPSIVAC. So Dr. Kamar, ultimately round and round, I have to come to you again for this. So final comments from your side on this, Dr. Kamar.
Okay, so essentially, as I showed you, that in the journey, we evaluated the dose of 0.1 ml for 3 days, 0.2 ml for 3 days, and 0.3 ml for 3 days. We have not gone higher than that clinical setting because we got desired results at 0.3 ml. But in a real world scenario, I mean, you would almost like to know what is a therapeutic window, what is the highest dose which you can use without significant side effects. So from that point of view, therapeutic window for this product is very high. And probably that's the reason somebody has already used 0.6 ml given intravenously instead of 0.3 ml for 3 days. He used the higher dose of double the dose. But I'm aware, and there are publications where I'm one of the co-authors, that it can be given as high as 5 ml. The current dose is 0.3 ml for 3 days, but the therapeutic window is so high that it can be given for at least up to 5 ml. That has been the experience. So, I mean, if you want to judge in the critical conditions, and what you can do is you can titrate the dose upwards if you are not getting desired result, as long as they fit into those criteria. So that is one of the way of personalizing the medicine for your patient per se. But as a thumb rule, we will recommend that start 0.3 ml for 3 days and then evaluate and see which way the patient is moving. But there is nothing wrong in repeating the dose or extending the days instead of 3 to 5, 6, 7, or increasing the dose or repeating it. Whenever, see, I have seen in a clinical setting where the patient becomes stagnant after a time, and then in those few of those patients, it was repeated after 7 to 10 days, and patient again started recovering. So these are real life experiences which cannot be done in a controlled clinical trial.
Thank you, Dr. Kumar. Dr. Kumar, my next question is, what I'm getting is a very simple question. It says, can SEPSIVAC be given to a pregnant or a breastfeeding women? So one is data, other is experience. So before I go to data, I'll ask for the experience of any of the panelists, Dr. Shweta, Dr. Rajat, or Dr. Ansari, anybody. Do you have any experience? And then I again go back to Dr. Kumar with some data on this. Can this SEPSIVAC be given to pregnant or breastfeeding lady?
So we have not used SEPSIVAC in pregnancy, so I can't comment upon that. The other panelists and Dr. Kamar can probably share the data for that. So on ICU, obviously no breastfeeding patients were there, so it was not the patient were not given on that. But yes, we did specifically excluded pregnancy in patients who are given SEPSIVAC.
I think I would like as per the product insert, I think it is still not recommended below 18, and pregnancy and lactation was one of the areas where there was a debate. and any previous florid tuberculosis would also be probably a relative contraindication. So I would like to hear from Dr. Kumar, what is his take on this?
Okay, let me start from reverse order. In a tuberculosis, this drug is helpful, not contraindicated, because tuberculous bacilli is essentially intracellular organism, and we have conducted studies. A couple of them are published also, wherein it has been shown that giving SEPSIVAC will improve sputum conversion rate, and then in a control studies, sputum conversion is preponed by 4 weeks out of 8 weeks. So if in a control trial, if a parallel group reached a particular sputum conversion rate at 6 weeks, the investigational group reached at 2 weeks the same amount of sputum conversion. When you have a 100% sputum conversion in 8 weeks, in a UVAC arm or MW arm or SEPSIVAC arm, it reached at 4 weeks. Okay, so there's a 4-week preponement of sputum conversion in sputum positive tuberculosis. The dose used in those situations were little different because it's a chronic condition and not an acute condition. So it was used as every fortnight for 2 months, and it has also been found useful in India tuberculosis for a reconversion, faster reconversion, improved sputum conversion rate over time. So this is the background information in relation to tuberculosis. So any infection is not a contraindication to use SEPSIVAC. Okay, whether it is a gram positive, gram negative, it is not contraindication. Tuberculosis is a gram positive organism. Only thing is the immune dysregulation, which you see in gram negative sepsis, is of a different variety than what you see in a gram positive sepsis. And that's the reason we took a conscious decision to develop it for a gram negative sepsis and avoided using it in gram positive sepsis, but it's not a contraindication. Bacterial clearance will still be faster, but the immune dysregulation in a gram positive sepsis is different than gram negative sepsis, and this mimics more of a profile of a SEPSIVAC, and that's the reason we continued developing it for a gram negative sepsis and COVID-19, but not for gram positive sepsis. But it's not a contraindication. Coming back to the age, at what age you can start? I mean, if you, go back to our basics, immunology, we are born with innate immune system, and adaptive immune system comes little later. So there's no contraindication to the age. As far as I know, it has been given to 1 month old till the end of life. And only thing which you see across the whole age group is a local injection site reaction, which happens in 15 to 30% of the people. Most often it is self-limiting and you don't have to do anything except reassure the patient in advance. Okay, now coming back to pregnant and lactating women, as it is, any innovative product will start with the usual standard criteria where you restrict your age group to 18 to 65, you restrict childbearing female, not using contraception, you restrict it to pregnant female, you restrict it to breastfeeding female because of abundant precautions. But outside that, it has been used in pregnant women, it has been used in lactating female, and so far nobody has reported any adverse event. And knowing basic fundamental immune mechanism, it is not likely to affect them, because it does only thing it does is it enhances your innate immune response, which happens through macrophage and NK cells, which does not pass through placenta, which does not pass through milk. So the effect remains on the other side of the barrier. It does not cross the barrier. So this relative contraindications are true for the vaccines where the antibodies cross the barrier.
So, Dr. Kumar, indirectly, if we have to say, it is not contraindicated. However, as an indication, though we do not have an organized trial or study, but a lot of medical fraternity have used and they have not come across any side effects. So yes, it may be used in a simple language if we have to put it like this. But
Use
are
your
we getting a lot of questions?
in this exceptional procedure, use your discretion and weigh risk versus benefit, because there are still unknowns. While we know the science, we have some experiences, but we do not have a whole lot of experiences in this group.
Yes, thank you very much. So there are a lot of questions, Dr. Kumar, and some questions are like, even though simple but complicated, there's a very simple question which says, does SEPSIVAC reduces oxygen requirement? It's a simple one-liner, but we have a simple response to this.
Clinician should answer that, but the short answer is yes.
Yeah, so this is to my panel. This is to my panel. I think Professor Benedicto doesn't have an experience, so I cannot ask him. But definitely, or maybe if I put it in a different way, Professor Benedicto, since you've been using other immunomodulator like TOSI and all, so any of the drug, do you see reducing oxygen requirement? And then I'll come to this question of using SEPSIVAC reduces oxygen requirement to our other panelists of Dr. Ansari. Professor Benedicto, first to you.
Yes, we do see reduced oxygen requirements of patients being reduced once we hit that sweet spot of, let's say, giving possibly TOSI, and of course starting the steroid early. But of course, it's not consistently being demonstrated. I mean, some patients would tend to benefit more compared to some other patients.
Thank you very much. Dr. Shweta, would you like to add something here with your experience? Or would you like to respond to this?
Yeah, definitely. So we have used in two type of patients. One is one group was patients who were on high flow oxygen therapy. So we have seen good effect, like the decrease in the oxygen requirement in these kind of patients. Patients who were already on ventilator, a few patients definitely showed decrease in the oxygen FiO2 requirement, but the patients who did not show the FiO2 decrease, there are various confounding factors. Once the patient is already on ventilator, his lung parenchyma is quite destroyed. So, and once he is on ventilator for more than 48 hours, there are chances of developing VAP in this patient, which again reduces the oxygen levels in the patient. And there are many other bacterial infection altogether. The multi-organ dysfunction can lead to decrease in the oxygen levels of the blood in the patient. So what we have experienced is the patient who are not on ventilator and who may go for a ventilator. If we use SEPSIVAC in these kind of patients, there is definitely decrease in the requirement of oxygen, and the rate of these patients going on ventilator was less. Recently, I have also used IV SEPSIVAC in few patients, almost 7 to 8 patients, and what we have seen in the 6 patients who were on HFNC, we did not require ventilator. 2 patients though required ventilator despite of SEPSIVAC.
Thanks. Dr. Rajat, any pointers from your side?
I think I agree with Dr. Shweta and other panelists that yes, the SEPSIVAC when used early has shown improvement in oxygen requirement, and we also found that the improvement in PF ratios and weaning was less shortened from ventilator. So that proved a significant improvement, especially in patients with pneumonia. So that was the positive aspect of SEPSIVAC IV which we used.
Thank you very much, Dr. Rajat. Dr. Ansari, you had been off the screen with little abracadabra, but you're back. So we brought you back on the screen for the comments, especially on oxygen requirement.
So if you go back to our study, our own experience, I think 75% of our patients with the standard care and with SEPSIVAC did show a clinical improvement from severe COVID-19. So I can only say that as an adjuvant given on a timely basis to severe COVID-19 patients, it definitely increases the chances of your recovery. But again, to say that whether it is alone agent to reduce oxygen, I don't think it directly acts on the organ. So it is an indirect effector of improving your immunity and giving you a better immune response in conjunction with other established therapies like oxygen therapy, your non-invasive support, and your antiviral and other immunomodulatory agents, especially steroids. So I would only say.
Thank you very much. Thank you very much, Dr. Before we close, one final comment, Mr. Sisodia, I'm getting certain comments from our partners, and would you like to give them some confidence? There are partners from Indonesia, Malaysia, Myanmar. I'm getting a lot of comments. How exactly availability you said is going to be there, and then how they can partner with us, Philippines also quickly. How quickly we can go through the FDAs and MOH and all. Would you like to comment finally on this?
Yeah, so 2 pointers at this point in time for us. As I said earlier, one is about the regulatory part of it, just to get the permission to import and distribute the product. And second would be about continue to talking to the medical fraternity. We had very good interaction today. All of us got benefited from it, and we need to continue building up this on individual basis also, as well as in the groups. So based on these 2 parameters, I think we should be able to progress, as I told you, within few days, and look forward to play a role in coming days to alleviate this tragedy which we are all going through in different parts of the world. Yeah.
Thank you very much. Thank you very much, Mr. Sisodia. And I would very, very gratefully thank all of you, all the panelists. Dr. Shweta, you've been wonderfully, talking to you. You got free in between. Dr. Professor Benedict, I'm sure you've got few pointers and whatever other questions. Anytime we can meet. Dr. Kamar, your baby is going to grow and it's going to go to many countries. I'm sure Philippines is going to be very quick on this. Professor Rajat, I understand you have to leave for another session at 6:30, and thank you very much for being, accommodative and being part of this panel. Dr. Ansari, all the very best. Mumbai rocks, you rock. I think it's a wonderful panel. We all are so wonderful and beautiful. We are going to get many more questions, sirs and panels and everybody. And I'm sure, we will stay in touch with you for the responses. And we thank you very much to all my audience, who have put in wonderful questions and taking out time for this webinar. And I'm sure you gained something out of it. And I wish all the best to Philippines, especially to the medical fraternity, for coming out of this COVID situation. India has come out from the second wave, but it's not going to go anywhere. And I'm sure this SEPSIVAC is going to help you. Thank you very much once again to all of you, all my panelists, all the audience, everybody who was part of it. And now, thank you very much to all of you. And I hand over this floor to Pauline and the team sitting on the other side who made it possible for me. Thank you very much once again to everybody. Thank you. Pauline, over to you and the other colleagues, other partners from Philippines. Thank you once again.
Thank you, Dr. Vipin.
Thank you. Thank you, Dr. Vipin.
Thank you very much, Dr. Sethi. And I would like to call in the sales and product managers of Biocare Life Sciences, Mr. Mike Pere and Mr. Mark De Vera.
Hi, thank you, Ma'am Pauline. So thank you, Dr. Vipin Sethi, and to all the doctors who stayed on. Welcome to the quiz but fun segment. We'll have some questions that will allow you to win a prize. The mechanics are easy. The flash on the screen, and all you have to do is to write your answer in the question box. The first person to send the correct answer after the timer stops will receive one of these about a personal choice question. Just a reminder to write your answer in the question box. All right, Sir Mike, let's get it on.
Thank you, Sir Mark, and good evening, doctors. Here is the first question. What is the recommended dose and administration of SEPSIVAC? Letter A, 0.3 ml given intradermally in 3 divided doses of 0.1 ml each on 3 different sites daily for 3 days. Letter B, 0.3 ml given intradermally in 3 divided doses of 0.3 ml each of 3 different sites daily for 3 days. Letter C, 0.3 ml given intradermally in 3 divided doses of 0.2 ml each of 3 on 3 different sites daily for 3 days. Or letter D, 0.2 ml given intradermally in 3 divided dose of 0.2 ml each on 3 different sites daily for 3 days. So timer starts now. Yan. And the correct, let us show the correct answer. Letter A, SEPSIVAC is given 0.3 ml, given intradermally in 3 divided doses of 0.1 ml each on 3 different sites daily for 3 days. And the winner is Dr. John Kathleen Domingo. Thank you. Congratulations, Doctor.
Congratulations,
You just win
Doctor.
a power bank.
Power bank. That was a good reminder, Sir Mike, on the recommended dose of SEPSIVAC. So, doctors, here's another question. Which of the following statement on sepsis is true? Letter A, sepsis is a life-threatening organ dysfunction due to dysregulated host response to an infection. Letter B, in COVID-19 sepsis, the infection is the virus and the life-threatening organ dysfunction is all the organs that can dysfunction, including lung, brain, kidneys, and heart and liver. Letter C, COVID-19 that causes organ dysfunction is viral sepsis or all of the above, or letter D. Key in your answers now. Time is up, and the answer is letter D. And the first person to get the correct answer is Doctora Milanen Flores. Congratulations, Doctor.
Congratulations, Doctor.
You won a personal air purifier.
Moving on, here is the third question. Aside from SEPSIVAC Mycobacterium W injection, Biocare is also marketing other COVID drugs. Letter A, remdesivir. Letter B, tocilizumab. Letter C, favipiravir. Or letter D, all of the above. Timer, please. Okay, all right. Let us see the correct answer. And the correct answer is letter D. Biocare is marketing remdesivir, tocilizumab, and favipiravir. And the winner for this question is Dr. Joseph John Biel. Congratulations, Doctor. You won a power bank.
Doctors, there are a couple of more prizes to give away at this time. We have a trivia question. Who is the Cuban boxer that upset Filipino legend Manny Pacquiao in the recent fight in Las Vegas? Letter A, Yordonis Ugas. Letter B, Lourdes Ugas. Letter C, Conor McGregor. Or letter D, Conor Gregor. Key in your answers now. All right, let's show the audience the correct answer. Letter A, Yordonis Ugas. And the first person to send in the correct answer, Dr. Dino James Abong. Congratulations, Doctor. You won a personal air purifier.
Congratulations, Doctor. And finally, we are down to our last question, another sports trivia. Philippine weightlifter Heidelin Diaz won the country's first ever gold medal in Olympic Games. Who is the first Olympian medalist for the Philippines? Letter A, Manuseto Velasco. Letter B, David Nepomuceno. Letter C, Teofilo Ildefonso. Or letter D, Leopoldo Cervantes. Timer starts now. Okay, and the correct answer is letter C, Teofilo Ildefonso. And the first person who got the correct answer is Doctora Rodora de los Reyes. Congratulations, Doctor. You just won a bank.
Congratulations, doctors. May we request for all the winners to please write your name and address on the question box in order for the tokens to be delivered to you.
So we hope you enjoyed this quiz segment. And with that, let us call on our business unit head for Biocare, Mr. Ronald Palacio.
Thank you, Mike and Mark. Hello, doctors, and good evening. Thank you for taking the time to join our webinar and SEPSIVAC product launch in the country, a first in the world innovation for sepsis management. It is our hope that the topic we presented gave you more insights on the management of COVID that is relevant to your practice. On behalf of Biocare Lifesciences Incorporated, I would like to thank our speakers, Dr. Abdul Samad Ansahi and Dr. Yatin Metha, for sharing with their experience on the use of SEPSIVAC in the COVID cases. Our gratitude as well to our panel of reactors, Dr. Rajat Agrawal, Dr. Shiveta Chandakedi, and Dr. Jubert Benedicto, and to our moderator, Dr. Bipin Sethi, for moderating tonight's forum. To our partners agency, MedConcept, thank you for organizing this webinar. Doctors, SEPSIVAC will be available in the Philippines through Compassionate Special Permit. As a final note, we would like to request our participant to answer a brief survey before you leave the webinar. On your screen is a QR code and link, so please scan the QR code and type the link on your browser, whether it's Google or Safari, and begin answering the survey. Once you have accomplished the survey, you will automatically receive your e-certificate, which you can download. You will also receive a copy of your e-certificate to your email address. The survey is open from 7 to 11 p.m. only, so don't forget to accomplish the post-webinar survey form so that as to facilitate your certificate of attendance. Once again, thank you, doctors and healthcare partners, for spending your evening with us. Stay safe, everyone.
Biocare Lifesciences Incorporated is a pharmaceutical company engaged in the distribution, importation, registration, and marketing of various therapeutic products across the Philippines. We at Biocare commit ourselves to service excellence in the promotion and delivery of essential as well as technologically advanced medical products that are within reach of the average Filipino. We pride ourselves in our company core values: punctuality, integrity, commitment, accountability, and this reflects on our service. Our vision is to play a significant role among the pharmaceutical companies in the Philippines, delivering high-quality yet affordable products and services that improve the quality of the Filipino lives it touches. We are dedicated to our mission to be among the top market leaders in our chosen segments by providing qualitative and affordable medicines to the community and year-to-year growth of employees and stakeholders, as well as to develop a young, talented, and experienced workforce. In 2011, Biocare Lifesciences Incorporated was founded by Managing Director Dilip Tiwari and President Kimberly Chua Tiwari. By 2012, we started the distribution of our products to numerous multinational and national companies. In 2013, Biocare registered over 23 products for different segments, and by 2015, we expanded our staff, deploying our new sales force to 17 areas in Central Luzon, South Luzon, and the Visayas region. In 2016, the expansion of the sales force and marketing team continues to the Greater Manila area, Visayas, and North Luzon, and we've moved to the new HQ with a complete rebrand. By 2017, we successfully launched Cefsul, a combination of third-generation cephalosporins and beta-lactamase inhibitor antibiotic. Now, Biocare expands to Cambodia and launched our first inhaler line with salmeterol fluticasone, Salflosol MDI, and Highcare, brand of salbutamol. Biocare has products from reputed pharmaceutical manufacturers around the world whose facilities are approved by various regulatory boards. And Biocare hasn't even reached its full potential. In 7 years, we've only laid a foundation. Soon, we'll strengthen our reach to the Mindanao region and eventually spread to the whole Asia-Pacific region with a culture of leadership and accountability. Looking forward, we want Biocare to be a corporation that you can always trust. Providing quality, defining life.
whenever you're sad. Carry on when your breeze is down. All I do is grow old with you. Anything when your tummy aches, feel your so it could be someone else growing up with you. Miss you, kiss you, give you my love and you are need you, need you, even when you hold out of control. So let me do the dishes on the kitchen sink. You had too much to drink. Oh, I could be the girl who grows up with you. Kiss you, give you my love when you have need you, feed you, even if you never could. Dishes in our kitchen sink. When you had too much to drink, so it could be so nice. I know with you. You smile whenever you're sad. Carry around when your face is bad. All I wanna do is grow.
Ending webinar in 3, 2, 1.